So today I will start with glycogen metabolism. Let us see from one of my old notes what glycogen metabolism is. So as you can see here, just hold on a second. I'm just clicking on editing, commenting. So we will be talking about glycogen metabolism. So you know that glycogen, it is the storage form of glucose in animals, and it is stored in the liver 6 to 8% and skeletal muscle 1 to 2%. So the quantity of glycogen in muscle is around 250 grams, and that in liver it is 75 grams. That is because muscle mass of liver is less than that. So muscle mass is greater than that of liver. It is stored as granules in the cytosol. So if you see the cell like this, you will see that there are small glycogen granules present. And in the cytoplasm, all the enzymes of glycogen synthesis and breakdown are present. Just hold on. হ্যালো, হ্যাঁ হ্যাঁ বল, হ্যাঁ বল। না না ঠিক আছে বল। হ্যাঁ, অনলাইনে দেখতে তো পারো, অসুবিধা কিছু নেই। ওই দিন কলেজ খোলা আছে। হ্যাঁ নিশ্চয়ই, হ্যাঁ কলেজ খোলা আছে। মানে পিঠে থাকলে এসো না, কিন্তু কলেজ খোলা আছে। হ্যাঁ তাহলে তোমাকে আসতে হবে। আগো কি হচ্ছে? হ্যাঁ হ্যাঁ হ্যাঁ, ঠিক আছে ঠিক আছে। ওকে ওকে, না না ঠিক আছে। হ্যাঁ না, ছুটি নেই।
টফি।
হ্যাঁ হ্যাঁ, ওকে ওকে, হ্যাঁ তাই করো। কিন্তু ওই দিন ছুটি নেই, তোমার অসুবিধা আছে সেদিন, সেদিন আর কলেজে আর ক্লাসটাও নিও না সেদিন, তেমনি ক্লাসও তোমাকে তো নিতে হবে না। তুমি প্লেন অ্যান্ড সিম্পল এসো, চারটে দেখে টেকে তারপরে ক্লাসে চলে যাবে। হ্যাঁ হ্যাঁ হ্যাঁ। এখন কি অবস্থা, টিচাররা ছুটি নেওয়ার জন্য আমাকে ফোন করছে। যাই হোক, আচ্ছা আমার কথা শুনতে পাচ্ছ তোমরা?
Yes, sir.
Yes, sir.
Yes, sir.
Okay. So the what is the function of, the question will come, what is the function of liver glycogen? Basically to maintain blood sugar levels. So when your blood sugar levels are falling, when your blood sugar levels start to fall, then there is a glycogen breakdown starts to take place. I'll open one of these, my whiteboard. So when blood sugar level or blood glucose levels start to fall, then in the liver there is glycogen breakdown into glucose. So blood sugar levels stabilize. This happens during fasting. During fasting or night when you are sleeping. So when you are doing your roza during Ramzan, at that time actually the whole day, what happens is this process, glycogen breakdown, keeps on taking place. Breakdown takes place and your blood sugar levels are maintained. Levels are maintained. What happens normally when this process of gluconeogenesis takes place during the night, this takes place during the day. Then after that 6 o'clock or so, when you start eating, you normally, it's better preferred to break fast with something like dry fruits and nuts, so that you get something like fructose, fat and protein. You immediately don't take glucose water, because it is not needed. So this is actually what happens during and liver stores glycogen in a well-fed state, and they are depleted during fasting, which I told right now. Muscle glycogen, they serve as fuel reserve for the supply of ATP during muscle contraction. So, muscle glycogen, the fuel reserve. For ATP generation. So, glycogen, they break down into glucose and glycolysis. You have ATP generated. They also, this ATP also, they fall short. At that time, creatine phosphate. This starts to give energy. So there are a question comes, why glycogen is chosen instead of fat as the first choice of fuel reserve. So your fat will not be metabolized, it's your glycogen which will go. First reason is it's very rapidly mobilized. Glycogen can be easily mobilized. Then glycogen can generate energy in the absence of oxygen. You don't need an aerobic condition. And the brain requires a continuous supply of glucose, which mostly comes from glycogen, because brain cannot take other things. It needs only exclusively glucose. Brain relies exclusively on glucose. And glucose can flush because there is a blood-brain barrier present. Blood-brain barrier is present. It only allows passage of only selected substances are allowed to pass. So, when a choice comes, when a person is starving, glucose reserves are diminishing. Glucose reserves are diminishing. At that time, brain gets the first call for glucose. At this time, let me tell you the first enzyme in glycolysis, that is hexokinase. This converts glucose to glucose 6 phosphate. This is present in high concentration in brain. This enzyme. Right? In high concentration. At the same time, in liver, hexokinase is not present. What is present in liver is glucokinase. And what is this also does the same reaction. Glucose is converted into glucose 6 phosphate. So only one difference. Affinity of hexokinase enzyme for glucose is much higher than that of glucokinase, approximately 50 times. So whenever the glucose comes, the first call for glucose gets from the brain. If brain has got sufficient glucose, then it goes to the liver. Because the hexokinase enzyme, which is present in high amount in the brain, it will pick up this glucose very fast. এটা বুঝতে পেরেছো? Is this clear?
Yes, sir.
Yes, sir.
Yes, sir.
Why brain gets the first call for glucose? This is one biochemical reason. And as I said, that first call is glycogen move, because it is very fast. Mobilization of the fat is slow and it needs oxygen. And it cannot produce glucose to a significant extent. You can call fat as a fixed deposit and glycogen as savings deposit in the body. Savings, like in a savings deposit, what you do? You need 200 rupees now, you go to the bank and then it's safely placed stored over there, your money is stored over there. You take 200 rupees out from the bank. Right? And this you use for your work. But a bulk of money you keep as fixed deposit that you want to save for your future. Everyday use, you keep your money in your savings deposit. But for a long-term interest generation, you keep it in a fixed deposit. So in terms of emergency, when you're dieting or something, when your glycogen reserves have gone very low, at that time, this fat is broken down. So the first term which we will talk is glycogenesis. So it is, as you know, it is the synthesis of glycogen from glucose. It is glycogenesis. Synthesis of from glucose. When glucose, lot of glucose is present, it is converted to the form of you're generating glycogen. are generating glycogen, that is glycogenesis. So when you have taken a lot of food, lot of sweets and all this sugar gives some energy, but the excess energy will be stored as excess energy in the form of glucose as glycogen. You can say that it is stored as glycogen granules in the cytoplasm. Basic structure of glycogen, you will see it's several units of these glucose, they go suddenly, then you will see a branch like this. Then again, this goes, you will see another branch like this. Then another branch like this. So in glycogen, say this is one glucose, and this is one glucose, this is another glucose, this is another glucose. They are joined together. This type of linkage I told, this is alpha 1 to 4 glycosidic linkage. So you have this glucose alpha 1 to 4 glycosidic linkage, another glucose alpha 1 to glycosidic linkage, another glucose alpha 1 to 4 glycosidic linkage, another glucose glycosidic linkage. So like this, these are all alpha 1 to 4 linkages. You also have branching points. And this branch point is alpha 1 to 6 glycosidic linkage. But then these again are alpha 1 to 4. Is it clear? Yes or no?
Yes, sir. Sir, only branching points are having 1 to 6 linkage, na?
Only
That way.
branching points.
Okay, sir.
It would have been better if I had drawn this like this. These are all, but I have not drawn it just for simplicity. This is alpha. Branching point is only alpha 1,6 glycosidic linkage. So structure of glycogen is similar to starch. Starch also has got branch point. Starch also has similar branch points. But there are two differences. Number of branch points in glycogen is far higher than that of starch. And glycogen is found in animals, starch in plants. Found in animals. It is the storage carbohydrate. And starch found in plants. Also storage. Alute থাকে. And where is the exceptions? Fungi have got glycogen. If you consider fungi as plant. And in animals, Ascidia. They are a group known as tunicates. Their test is made of they contain starch. Similarly, you can find cellulose in plants, chitin in animals. Same exception. Fungi have got they are chitinous and they have got cellulose. Next, we go to the next part. Are you able to see glucose here?
Yes, sir.
Yes, sir.
Glucose in the liver, we are talking glucokinase. There is no hexokinase. ATP is made into ADP, and glucose kinase does the same thing like hexokinase, but in the liver becomes glucose 6-phosphate. Then the enzyme phosphoglucomutase what it does, it converts glucose 6-phosphate to glucose 1-phosphate. In the first carbon, the phosphate goes. Then UTP, uracil triphosphate, is hydrolyzed. And you have UDP glucose. The name is, enzyme is UDP glucose pyrophosphorylase. And you make UDP glucose. Then this UDP is removed. This is known as another enzyme, glycogen initiator synthase. You're trying to synthesize. So, first glycogen, second glycogen, the first glucose, second glucose, third glucose. It comes. This is, say, for example, there is a base. You need a base of a glycogen chain. You cannot start from zero. You need a primer. There is this OH group. To this OH group, it becomes O. So, it's the glycogen chain, and there is an OH group. That H goes, and to that another bond is formed, and gradually these you need a trimer actually. You need 3 residues of glucose, where at least minimum to start this glycogen synthesis, you already need to have something like, you need to have something like, right here better. Okay, this is the base. OH. So you really need to have something like one residue of this glucose. UDP has gone out. To start. So the second thing is this line, requirement of primer to initiate glucogenesis. So you need a small fragment of pre-existing glycogen that needs to be present as a primer to initiate this glycogen synthesis. So in the absence of glycogen primer, a specific protein called glycogenin can accept glucose from UDP glucose, the OH of glycogenin. So this you can call glycogenin. Or you can already have at least a small fragment of small fragment of glycogen must already be present. And to this fragment, all new from UDP glucose, all other glucoses are new addition of glucose. On UDP glucose. So far clear? Is it clear?
Yes, sir.
Yes, sir.
Yes, sir.
ठीक है. Now you can talk about 13 UDP glucose glycogen, then the famous enzyme. After this is done, like initiator synthase work, a new enzyme, the glycogen synthase. Now what it does, the 13 residues come and you have all these alpha 1,4 cleavage taking place. So basically UDP glucose comes, this is say UDP glucose. This is the glucose molecule. So 13 molecules of this come and it releases 13 UDP and to this 13 glucose are added and the bond bond. This is alpha 1 to 4 glycosidic linkages. First, you had the priming and initiator, glycogen synthase, and then this 13 for continuous addition, you have the main glycogen synthase. Synthase, some people say synthetase. Yes, so you have this alpha 1 to 4 bond. Now, branch points need to be created. Formation of branches. So the enzyme name is glucosyl alpha 4 to 6 transferase. Right? This enzyme. This enzyme needs. Achha, can anybody of tell you if the screen went blank for one moment? Did the screen go blank?
No, sir.
Just for a second or so?
No, sir.
No, sir.
Okay. So that means for me it went. It means there is a loose connection between my monitor and the CPU. I'm using my desktop. Holidays I use desktop. Find it more comfortable. So this needs this enzyme glucosyl alpha, but this needs at least 5 to 8 glucose from the non-reducing end. So at least 5 to 8 glucose residues. Residue is one of what? Just one of this is a residue. Residue must be added before the 1 comma alpha 1 comma 6 glycosidic bond is formed. Is it clear?
Yes, sir.
Yes, sir.
Yes, sir.
Then we can proceed to the next part. So, like this, elongation starts to take place. And after 5 to 8 alpha 1 to 4 glycosidic linkage, what you have is you have one more 1 to 6 glycosidic linkage for the branch point being made. So branch points, this is the enzyme branching. So this is how glycogen is synthesized. Now we will be talking about how glycogen is broken down. So glycogen, it is the degradation of stored glycogen in liver and muscle. This is glycogenolysis. So you know when it takes place during starvation, when your blood glucose levels fall. Also, I can tell you when you are studying, suddenly you become a huge amount of glucose is taken away. You don't lose fat, but you lose a lot of glycogen. During a studying or high brain activity. A lot of glucose is consumed. Consumed. This is just for curiosity purpose. So let's see what happens in this case. বুঝির কিছু লিখলো দেখি। পার্ডেন করতে বলছে, ঘুম পাচ্ছে শুভজিতের, 4% হয়ে গেছে। Okay, so this is a 4 units of glucose. This is the glycogen molecule. This is the branch point as mentioned here. Right? The enzyme is glycogen phosphorylase. It keeps on breaking them into maltose, maltotriose, and glucose 1-phosphate. Glycogen molecule broken down into first is maltose. Maltose is 2 glucose units attached by alpha 1 to 4 glycosidic linkage. Maltotriose, that is, it has got 3 glucose units. 3 units alpha 1 to 4 glycosidic linkage. And glucose 1-phosphate. Since the phosphate is always at the first carbon, you have the phosphate here always attached. Now what happens? So you have 3 units, 2 units, and 1 unit. These small little compounds that is broken down. And you will have the branch point also existing. You have the branch point also existing. So this is what you have. The people call these things, sometimes they are known as limit dextrins. So let us now get back. Glycogen phosphorylase makes these things and glucose 1-phosphate, this plus this plus the two things, these three things are formed. So the resulting is you get this limit dextrin present. Now you have a debranching enzyme. You are breaking it down. It has got a transferase activity. What it does is these top three of this is transferred to this end. And this becomes longer. The branch point still exists. Then this debranching enzyme has got alpha 1 to 6 glucosidase activity and it removes the glucose. And this becomes a straight chain again. Again, glycogen phosphorylase enzyme, it breaks it into glucose 1 phosphate. So this is how glycogen is broken down. Am I clear?
Yes, sir.
Okay. Then I end my class here. This is the glycogen metabolism.
Thank you, sir. It was so close. Only 1% charges left. Thank
Okay,
you, sir.
okay, great. Okay. So any questions? Abha, questions?
No, sir.
Anybody else? Do you have questions?
No, sir.
Okay. I will send you the recordings and please keep on reading these chapters. I tell again and again. They are going to, otherwise it will be a problem. So
Sir.
right now, perhaps until New Year, we will have all our classes online because I will be going to Calcutta on the 25th, and this particular week, let's see if I can manage it. Maybe on the 24th, I can take one of your class offline, and then we will continue our classes online. And then in January again, when it reopens, we will again start offline classes again, offline online mix. Our studies should not stop. Okay? So next Wednesday তে হবে না ক্লাসটা, ঠিক আছে?
স্যার আমাদের থার্সডে থাকে স্যার।
তোমাদের থার্সডে তো? ও
হ্যাঁ।
আচ্ছা, তাহলে থার্সডে তো weekend, that is 23rd, না?
Yes, sir.
Okay, then we can think about offline class. Offline, we can have that offline class. But then the next 5 days after Christmas, that will become online. Okay? Because the New Year, I don't want. I want to go back to Calcutta for some days, and therefore physically this house will be empty. So I'll be in my house in Calcutta. So let's see. We will have online classes from there. Okay?
Okay, sir.
Okay, sir.
Okay, I'm